Good afternoon, everyone. Thank you for waiting. And thank you for joining today. I can see that many people have joined, so I think we are ready to make a start. My name is Virginie, and I'm the Research Infrastructure Project Manager at EURORDIS. And we also have our two speakers online with us today. Professor Fulvio Mavilio, who is the Scientific Director of Généthon in France, and also Dr. Heidi Howard, who is a bioethicist at Uppsala University in Sweden. So just to tell you how it's going to work for today's webinar. So we will have two presentations. Each will last about 20 minutes and will be followed by a 10 minutes question and answer. We will take questions after each presentation, if that's all right with everyone, because it helps really with the smooth running of the webinar. And both of these presentations will introduce genome editing, but from different perspectives. So the first one by Professor Mavilio will explain the science behind this technology and the potential for future clinical use. And the second one by Heidi will focus on ethical consideration relating to the use of genome editing for clinical application. So let's start with the first presentation, which you can see now on your screen. So thank you, Professor Mavilio, if you want to get started.
Thank you. First of all, good afternoon, everybody. I hope you can hear me well and see me well. So my job today is to give you an idea of the science behind this new technologies for editing the genome, for doing gene therapy for rare diseases. As you can see in my first slide, this is just a very simple picture that shows you the basic principles of gene therapy. So what we do today, we do this in patients, of course. So gene therapy, as you know, today is a clinical reality. And there are two forms of gene therapy, one that you see here in blue and the other in orange. So the blue is called ex vivo, which very simple translation of this from the Latin is that you take cells, normally these are stem cells from the body of a patient, you do genetic modification, which today essentially means using a viral vector, so something that derives from a virus in which we put the therapeutic gene, and we use these things to put genes into cells. So once we have completed the genetic modification of cells, again, with the addition of a therapeutic gene, we put the cells, we transplant the cells back into the patient, and this is essentially the basis of the therapeutic intervention. This is applied today is essentially blood disorders for the reasons that stem cells, we know blood stem cells very well, we know how to get them out of the body, we know how to manipulate them, so how to use viral vectors to put genes into the cells. And we know extremely well how to transplant them back, because this is something that clinicians do since 50 years. The second system is called in vivo, which is probably the simplest to understand. We just use a viral vector again, although this is a different viral vector with respect to what we use for the genetic manipulation of stem cells, and we inject it directly into the patients. Directly means it can be in a very limited place, like for instance in the eye for correcting severe blindness, or systemically, which means we put it into the blood and we allow these vectors to reach all the organs. And this is something we do for treating systemic disorders, like for instance muscle diseases or coagulation factors diseases, so to put genes into the liver or in the brain, in the central nervous system. And we use the blood as a way to access these tissues. So this is gene therapy today. We use a vector, we use a therapeutic gene, we put this gene into the genome of the cells that we want to treat or of the organs that we want to treat. And there are many examples of success. I mentioned already hemophilia and blood diseases and so on and so forth. So what is the new technology that essentially is the subject of today's presentation about? This is called gene editing. So instead of adding a gene to a cell or to an organism, that's what we do with classical gene therapy using viral vectors. The purpose of editing, as the name says, is actually to correct a genetic defect. So not to add a functional gene in cells or organs that has a deficient function of this gene, like in most rare genetic diseases, but actually knowing what the defect is, correcting it directly into the genome. And when I say the genome, I mean the genome of somatic cells. And the second speakers will actually discuss the issue of what is the difference between correcting a blood cell or correcting the germinal cells, and so doing modifications that can potentially be inherited. So gene editing, and you see in this picture, okay, the picture in the center is obviously just to understand what we do. So that's the DNA, and the nice lady with scissors is trying to cut out bad base pairs and add the good ones. So this doesn't tell very much, and I will tell you how we do this, and we obviously don't use scissors. The pioneers who are shown in these slides of this technology, there are many fathers of gene editing, fathers and mothers actually, and you see some of them. George Church, who works at Harvard University in Boston, Feng Zhang from the MIT, again in Boston, Massachusetts, and the two ladies that really originated this field in terms of invention. These are Emmanuel Charpentier, French-born scientist, and Jennifer Doudna, California, who actually adapted some of this, the technology that I'm going to explain to you, and made it really very simple to do this kind of technology. Simple in the laboratory, not as simple to translate this into a clinical reality. We will discuss this at the end of my presentation. So what is gene editing? So I will try to explain what we can do and what all this is about. So you see here a piece of DNA and a little scissor. So what this means that we have tools, molecular tools, which I'm going to explain in a minute, with which we can introduce cut in the DNA of our cells. Now, why do we want to cut the DNA of our cells? Well, what we want to do is not just cutting. What we want to do is correcting, so editing a genetic defect. But to do this, we use a combination of tools that scientists invented and of systems that the cells normally have to repair their genome. So all our cells have mechanisms to repair mutations or to repair random breaks of DNA that, for instance, when we go sunbathing in the summer, the sun, one of the effects of the sun is to actually introduce cut in our DNA. Radiation damages the DNA. So all the cells have systems to repair. And that's exactly what we use to do gene editing. We use the repair mechanism of the cells, and we try to direct the DNA repair mechanism of the cells to actually correct mutations that cause disease. So this starts with a cut. So we use a molecular scissor to introduce a cut in a very precise place of the genome. So the technology part are these molecular tools that we use to cut the genome exactly where we want to cut it. What happens when you do this in a cell? Well, two things can happen. The cell can repair the cut by a system that you don't need to understand all these details, but just the philosophy. A system which is called non-homologous end joining, but it doesn't matter. Just the cell understands that there is a cut, and it rejoins the regions that are cut. In doing this, the cell introduces errors. So this sort of splicing mechanism is not precise. So if you want to kill a gene, so if you want to abolish a gene function, you use the system. You introduce a cut, you let the cells repair, and repair introduces mistakes, and you essentially destroy a gene by making mutation into a gene. This causes targeted, you can use the system to delete pieces of genes, to mutate precisely one specific gene. And for instance, for dominant diseases, what you want to do is actually destroy the mutations, the dominant mutation that causes the disease. Another thing that you can do is using another system that a cell has, they're both operating in our cells, which is called homologous recombination. Homologous recombinations means that when the cells detect a break, again, it uses the other chromosome. As you know, we all have 2 copies of everything in our genome. It uses the sister chromosome as a template to repair the damage. So this is a very accurate system that essentially copied from one chromosome to another and restore the existing situation. So we can use this system, we can harness this homologous recombination system by cutting the DNA and by providing into the cell another template, so not the other chromosome, but a new template that we introduce together with the molecular scissors, and we convince the cell to repair the cut with the gene that we have introduced. By doing this, we can correct point mutations, for instance, or we can correct small deletions, for instance, or we can correct most of the defects that are the basis of genetic diseases today, as we know them today. So this is what gene editing is about. Using a molecular scissor, allowing a cell to repair one way or another way. We have systems to convince the cells to do one thing or the other thing, and essentially correct a mutation or multiple mutations. So what are these molecular scissors? So this gets a little more complicated, but I will try to make it simple. So this figure shows you 3 types of molecular scissors that we have. There are 2 components that are important in all of them that you have to understand. One is that we don't introduce random cuts into the genome. We want to introduce precise cuts into the genome. So to do this, we use either proteins, so protein module engineered in the laboratory that are called zinc finger nucleases or tail nucleases. It doesn't matter what this means. What is important is that you understand the concept. Essentially, these are modular proteins that recognize specific sequences into the DNA. So since we know the sequence of the genome, we know where to go. We engineer protein that can detect specific sequences and introduce a cut. So these scissors have 2 components. One, which is the one that detects the specific DNA sequence and put the scissor into a precise place where there is the mutations, for instance. The other part, which is indicated by this little scissor, is the nuclease. Nuclease means it's an enzymatic function of this protein that introduces the cut into the DNA. So the enzyme cuts, and the other part of the protein, which is either a zinc finger or tailed domain, it's probably too complex to understand the difference, but as I said, it doesn't matter. They're essentially modular parts of protein that can read the DNA sequence and put the scissors where we want to put it. The last part of this figure, so the bottom part of this figure, is the most recent version of this molecular scissors. It's called CRISPR-Cas9. Very complex name, which is an acronym of an even more complex things that I myself never remember, so forget it. But what is important to understand, and this is the invention of Emmanuel Charpentier and Jennifer Doudna, is that this is not a protein that recognizes DNA, but it's actually a universal cut module, which is again indicated by the little scissor, that uses an RNA molecule to direct the cut to the DNA. So this originally comes from the immune system of bacteria. You've never heard about this, such a thing, and neither did I since until 5 years ago. But essentially, this system is extremely versatile because instead of having to engineer proteins, which are very complex machines to create in the laboratory, we very simply use a universal scissor, which we direct to just any sequence in the human genome by using a complementary RNA molecule. So this is extremely simple. any student in any laboratory can do genome editing by using this system. And this is why it's becoming so popular. I'm sure that some of you have heard about this CRISPR thing. And this is why any laboratories is now using this for all kinds of things, for engineering plants, engineering animals, changing things in cells, create preclinical models, or potentially clinical use. If we move on with this, of course, if you have questions, I'd be happy to look at this. This is just, let's say, for those of you that understands a little bit molecular biology, this is, you see the DNA, the double helix, and you see one of these molecular sisters in action. So this is a zinc finger nuclease. You will see all this colored module. There are in the arrows points to the zinc fingers, which are really fingers that attach to the DNA, recognize sequence, and direct the cut to a specific point. And then the blue part is which is indicated here as FOC1, which is a name, it doesn't matter, which is the actual sister that introduces the cut. In the other slide, so in the next slide, you see here, where is it? Oh, here it is, CRISPR-Cas9. So this again, it's a molecular sister, but you see here that the double helix of the DNA is open, and the way we guide the Cas9 molecule, so the sister, to the DNA is using this guide RNA, which is essentially a synthetic molecule that we make in the laboratory that reads the DNA sequence that we want to cut. So we direct a universal sister, so this is the same for anything, a plant or a human being, and we direct it with a guide RNA molecules, which is very simple and very easy to synthesize, and also very economical. So it's a universal system, very user-friendly, very powerful. What can we do with this? As I said, well, first of all, but just as for your information, this thing is so promising for a number of applications. I mentioned plants, I mentioned animals, so transgenic plants, transgenic animals, and I mentioned medicine, that a lot of investors are putting a quite substantial amount of money to develop this technology for medical use. You see some of those, so zinc finger nucleases, for instance, there is a biotech called Sangamo that invested hundreds of millions of dollars on this technology. And the CRISPR-Cas9, the one on the right, the newest technology, there are at least 2 biotechs that owns the major intellectual property in the field and that are investing hundreds of millions to develop medicines with this technology. I will just give you 2 examples of what we can use in medicine to correct diseases. The first example that I show you, as you see, is not a rare disease, but it makes very well the point on how powerful this technology can be. And it's actually gene therapy for AIDS. There are many ways of using this technology. One, for instance, instead of introducing cats to correct a genetic disease, is to actually abolish a function into the cells, as I explained at the beginning. So in the case of AIDS, as you know, the virus enters into our lymphocytes by using a receptor, which is a molecule that's expressed on the surface of the cell. Without that molecule, the virus cannot get in. So you create a cell that is resistant to viral infections by destroying the receptors for the virus. And this is what scientists in California are doing by engineering, you see it on the right part of the slide, by engineering the T cells, so the immune cells of the patients that are taken out of the patients. They used zinc finger technology to destroy the receptor for the AIDS virus. And then they put the cells back into the patients, hoping that this pool of engineered cells will be resistant to viral infections because the virus cannot enter into these cells. So this is a simple example of how, by destroying a gene, you can do something that is useful in medicine. For instance, renders immune cells resistant to viruses. The last slide and the last example I want to make is instead a genetic disease. This is something many of you probably know, is sickle cell disease. This is a mutation in the hemoglobin gene of the proteins that brings oxygen to the blood that causes cells, the red cells of the blood that you see on the left of this slide, the cells lose their normal shape and function and become sickled. So they have this shape and they become very rigid. When they become rigid, they clog the arteries. You see it in the right part, and so they cause all kinds of problems in patients: anemia, thrombosis, vascular damage. It's a terrible disease, and it is caused by one mutation in one gene, which is a simple mistake in one base in the 3 billion base pairs in our DNA that causes the hemoglobin to polymerize. Now, hemoglobin is normally a molecule that floats into the cells, if you look at the right part of the slide. And when it's mutated, it forms polymer that makes the cell rigid. So the red cells cannot squeeze nicely into the blood vessels. They become rigid. They clog the vessels, as I said. What can we do with gene editing? Correct that simple base change in red. So revert that base to the original one and change the sickling hemoglobin into normal hemoglobin. This is something that it's done these days in vitro. We can do this in the laboratory. We cannot do it yet in patients. But we hope that for this kind of diseases, we will soon be able to do correction also in cells and in patients. I finished my time, so I conclude my presentation by just saying, as I said, that this technology is extremely powerful, but we don't know yet the possible consequences of cutting DNA. There might be negative consequences of this. This is not studied well enough. There would be years before we understand what could be the negative side, the dark side of this gene editing technology. But certainly research is moving so fast that we will soon know what we can do well with this technology and what are the potential negative effects that we have to consider and possibly correct before using this into patients. Thank you for your attention, and of course I'm here to answer.
Thank you very much. Thank you, Fulvio, for this very informative presentation. So we are now going to start the Q&A in a few seconds. So there are two different ways to do this. So in a few seconds, you will see a microphone icon appear at the top of your screen next to the volume button. And you can either use the mic to ask a questions, or you can also use the chat box that's on the left of your screen. It might be good that when you don't speak, if you can mute your microphone, because otherwise there's an echo going on. So you should now all be able to see the microphone icon at the top of your screen. It should go green. And mute when you don't speak, and please use it if you want to speak and ask questions. Otherwise, you can also type your question in the chat box. So do we have anyone who wants to? We would like to hear your voice, so please don't be shy and use the mic if you want to. We could also, if no one wants to ask a question now, we can also start Heidi's presentation and keep all the questions for the end. Unless anyone... we have one question from Mathieu, and he asked whether we can mutate as efficiently intron and exon using genome editing. Can you edit maybe as efficiently intron and exon within the genome, Fulvio?
Yeah, that's obviously a good question. We can edit any part of the genome. So just to have you understanding, I made simple examples of mutation in exons, so in the coding part of the genome, so in the proteins, which are the basis of many genetic defects. But we can edit any part of the genome. When I mentioned briefly in one of my slides that we can do deletions, for instance, we can delete regulatory parts of the genome. So we can actually change, by doing gene editing, even gene regulation without touching the gene itself, but we can change the gene regulation. For instance, we can reactivate the synthesis of fetal hemoglobin in adult cells if this is therapeutic, and for instance for thalassemia or sickle cell anemia, this is a very interesting possibility. Or, as you asked, we can correct also introns. So if there are mutations in introns that alters the splicing of RNA, and that's the basis of the molecular defect of a specific disease, we can also correct introns. So we can introduce changes, small changes, large changes, large deletions, small deletions, into any part of the genome, as long as we have a tool, so a molecular tool, that can go exactly in that place. So, as I said, doing this with protein modules is a little more complicated. Doing this with RNA, guide RNA, is much simpler, and we can go in many more places with guide RNA than we used to go a few years ago with zinc fingers or TALEN proteins, just because this is much more versatile. But the answer is yes, we can edit anything in the genome: introns, exons, regulatory elements, whatever.
Thank you, Fulvio. We have another question from Ilke. Are the techniques used for genome editing for non-human animals, plants, are the same for the one used for human? And you use similar techniques to edit human and non-humans. And you also said these techniques are very simple. Is there a chance that this can be done in a private lab, from someone else's home, for example? It's true, I heard also that there's kits that you can get your hands on to try genome editing at home. What do you think of that, Fulvio?
Well, at home, I would say no, unless your home is really very well equipped as a research laboratory. Of course, manipulating cells and doing these things is, when I say simple, I mean simple for a research laboratory and personnel that is well trained. It might be even students, but you cannot do this in your kitchen. Just to let everybody understand that when we scientists say simple, we mean simple for research laboratories or for the biotech industry or for research hospitals, but obviously not from my grandmother in her kitchen, which does excellent pasta, by the way. On the other hand, what we use for modifying plants and what we use for modifying farm animals or to correct defects in patients is absolutely the same technology. DNA, as you know, is the same. It's the same molecule. All living organisms on this planet, at least, have the same molecular basis. It's this double helix of DNA that's made of 4 different bases. This is universal. So any living organism has the same basis for genetic information. So we essentially use the same tools. So we just change our molecular scissor to target a plant gene, resistance to cold or to parasites, for instance, or to target the human hemoglobin in the case of sickle cell disease. The technology is absolutely the same.
Okay, thank you very much. We also have another question from Danielle Renaud, asking if there's an option to use genome editing at very early stages of cell development, such as iPS cells for systemic diseases.
Yes, this is done essentially these days in iPS cells. And the reason is not because we know how to use well iPS cells in medicine. This is still in a field in its infancy, but because by doing modification into iPS cells or correcting genetic defects in iPS cells that come from patients with a certain genetic defect, we can create disease models in the laboratory. These are extremely useful. These are essentially advancing fields that have nothing to do with genetics. For instance, once we have, I make a very simple example. You have a patient with, for instance, spinal muscular atrophy. It's a disease. It's a terrible disease, and many of you may know. It's very difficult to find drugs, conventional drugs or medicine that can actually address this type of defect because we don't have models that we can use to screen for drugs. How do we do it? We can take cells from a patient with this disease, create an iPS cells with this mutation, and use these iPS cells to screen for hundreds of thousands of different compounds. I'm talking about chemical compounds, so the normal way of doing drugs, and find drugs that actually change the feature in motor neurons, for instance, that are the consequence of the disease. So this is an application that's enormously useful today, using genetically modified, genetically corrected iPS cells, or the reverse is also true. Create mutation into genes and understand their function, for instance. Into iPS cells, this is very useful. Many laboratories are creating an enormous amount of knowledge by editing iPS cells to create mutations and disease models, or to correct mutation and disease models, or to screen for drugs into particular diseases when animal models, for instance, are not available and we don't know how to develop our medicines. So there are many applications in medicines, which is not the classical form that you may think that I discussed of gene editing, so correcting the genetic disease, but actually using these as tools to develop medicines.
Thank you very much, Fulvio. I can see that Heidi had a question, but maybe we could come back to it following your talk, Heidi. So, and I also see someone is typing a question. If you hold on to it until the end of Heidi's talk, and then Fulvio will stay with us, right? Fulvio, until the end of the webinar. So you will have opportunity to ask both Fulvio and Heidi some questions at the end of Heidi's talk. So thank you, Heidi. If you want to start your presentation, please.
Okay. Yes, thank you so much for inviting me. So I'm just going to give a little introduction on ethical debates around new tools, very brief, and then I'll go straight into some ethical issues. So why do I want this little introduction? Because as Fulvio mentioned, a lot of the debate around gene editing has come about particularly because of this new tool, CRISPR tool, and it's a new tool. We had other tools before, just like I've put here on the slide, scissors. So you have scissors that can cut, or you could also have a laser cutter. And depending on which tool you use, you might have different concerns for safety, different concerns for accreditation, quality control, etc. You would not think of doing that for scissors in your home, but if you're using a more sophisticated tool, you might want to think about that. So that's a little bit simple. If you think of other tools where we've improved on our tools, here are writing tools, you would not have the same concerns about using a typewriter as you would have about using a computer. And again, I'm making this very simple, but it's just to get you thinking about, for example, privacy concerns. You're not worried about somebody hacking into your typewriter, but you might be worried about somebody hacking into your computer or your email or your cloud, wherever you're saving all of your data. So this is with every new tool, we might have some advantages, but we might also have some legal and ethical and social concerns. So that's the same thing with this new tool called CRISPR. And this happens when we study, for those of us who study the ethical, legal, and social issues of new technologies, whether it be in genomics or other fields, we have thought about this arc of the discussion, where a new tool becomes more efficient, it becomes usually cheaper to use and more accessible to more stakeholders. We tend to have this debate. One of the reasons for this is because if more people can use a tool, then we start worrying if they're using it in the way that it was intended, either practically, scientifically in the lab, are they using it well, or also ethically and legally, are they following all the laws? So that's one of the issues that's happening with CRISPR at the moment. So there's been a lot of use of CRISPR in the last more than year, but the debate has been going on for about the last 18 months especially. It's blown up a little bit in the academic literature especially. And again, the reason for that is CRISPR and particularly its uses. This is just recapping what Fulvio has said. It's been used in a lot of different organisms, non-human animals as well as humans, human cells, not in humans per se. And where that has brought us, or that has brought scientists, is that for the first time they are thinking, they're thinking, it's just a thought for now, that they could use this tool, this genome editing tool, in humans. So not just in human cells in a laboratory, but in humans. Actually take those cells and put them back in humans. And it's one of the first times that we've been able to think about doing that in a safe way. We've had other ways of doing it, as Fulvio talked about, classical gene therapy. But this gene editing tool is really opening a lot of doors. So that's why the discussion has blown up, or has taken so much space. And another reason that Fulvio also alluded to is the idea that we'd be able to use gene editing not just in somatic cells, so in your skin cells, but also in cells that you would use, others would use to create new life. So in your gametes, in your egg cells, or in your sperm cells. And that is completely new. So we had not thought of being able to do that safely in the past. So as I mentioned, there's a lot of different uses for gene editing, different microorganisms, agriculture, plants, insects, and of course a lot of model organisms, especially in the laboratory. So before I go on into the ethical issues, I just want to mention that overall, something to keep in mind is that there are a lot of scientific unknowns. It is a powerful tool, but there's a lot of things that we don't know completely about this tool. What would be the side effects? He mentioned it in passing, but the tool is not perfect. It could also target the wrong place and change DNA at the wrong place, so an off-target event, which you could argue could have, serious side effects. So we don't know the extent of these off-target effects. The other big issue, and that's why I'm talking now, is that this new tool is really challenging the ethical and legal frameworks that have been set up in genetics and genomics in the past, especially if you're going to try to target cells that are heritable. For the somatic cells, as Fulvio was discussing, like your skin cells or your blood cells, classical gene therapy laws and ethical guidelines tend to still work on that. But if you're going to talk about gene editing in your eggs or sperms or germline cells that will lead to this, then the ethical frameworks and legal frameworks are really challenged. And one last thing I want to say here before I go into specific issues is that for clinical geneticists and research geneticists and people like me who study the ethical, legal, and social issues of genetics and genomics, we've been used to for a really long time to consider genetics and genomics as a diagnostic tool or a predictive tool. And now with gene editing, we have to think about genetics as a therapy, as a treatment. And that's really, it might sound simple, but it's really a game changer for stakeholders in genetics and genomics, and we should keep that in mind because a lot of the ethical frameworks and the legal frameworks that have been set up for genetics and genomics look at genetics and genomics, and by genomics I just mean studying a lot of genes at once, look at genetics and genomics as a diagnostic tool. So you can see that just this flip from diagnostic tool to treatment could challenge a lot of what we've already set up. Okay, so now going into the ethical issues. First of all, going into the ethical issues, one size does not fit all. If you're going to have an ethical discussion that lasts more than 15 minutes, which is what I have left, you should really, really be specific about the context. And what do I mean by that? I mean that you should discuss specifically where you are talking about gene editing. Are you talking about gene editing in mosquitoes, or are you talking about it in humans? And there will be different ethical issues depending on this. So it's very important that you be specific about what you're talking about. And of course today is about human uses, but I also want to mention that the use of gene editing in non-humans will impact the environment and will impact humans in the end, so it's very important to keep an eye on them. especially since they're probably going to come to fruition sooner than gene editing in humans. So we have to keep a focus on that. But I will go now to the human uses. And again, to be context specific, you have to say, if you're having a discussion about the ethical issues, are you talking about the use in somatic cells, as the entire previous talk was about, or are you talking about gene editing in hereditary cells? So I'm going to separate those two, and I'm going to start with somatic cell gene editing, which has indeed a bit less ethical issues. It still has ethical issues, as gene therapy in the past also had, but we have thought about them a bit more. We've had time to think about them a bit more, and a lot of the issues have to do with safety. Is the technology safe enough to be used in the clinic in living humans, not just in their cells, in human cells in the lab? And so when we talk about safety, it's not just is it safe enough, but is it safe enough for the benefit that it brings? So that's a cost-benefit calculation. That sounds very technical, but actually there's a lot of issues there that have to do with ethics as well. And we would also have to go and think about what the acceptable uses are. You know, how severe does a disease have to be for us to consider the unknowns of gene editing and the benefits of gene editing? So the discussion gets a bit more nuanced there about the safety. And of course, this is true throughout all my slides. You know, who will decide on the criteria for this? This is a big issue. And who will decide for what disorders we could use gene editing and for what disorders we wouldn't use it? So we were pretty clear. A lot of stakeholders agree that we would use it for diseases. But then what about a person who would like increased muscle mass? If you have muscle mass that is under the normal, that could be considered disease. But what if you want increased muscle mass, let's say for increased athletic performance? Or what if somebody wants increased oxygen loading? You know, if we could ever do that, that would be enhancement, but you can imagine that in some persons it might be a disease. So this is a huge issue, disease versus enhancement use. Another pervasive ethical issue for all these new technologies is justice. Who will have access to this technology? Will it be everybody or only people who can pay for it? And you could discuss this in a country or in a continent like Europe, or you could discuss it through different regions, rich countries and low and middle income countries. And that becomes an issue if the disorders you're trying to treat with gene editing are more seen in low and middle income countries. Will the richer countries help them to get this technology or not? So I have no answers, but I have a lot of questions for you. So now on to hereditary and germline gene editing, which is where most of the discussion, the ethical discussion, has been, because this is really this is really pushing the discussion beyond what we understood before as gene therapy. So what I really want to stress here is that hereditary gene editing is gene editing in cells that could be used to create a new human in germline cells and or embryos is happening to a very little extent in human cells, in human cells in the research context. It's not happening in the clinic context. It's not happening now. But the debate is, what if it happened in the clinic? And that's why we're talking about this now. Okay? But I realize that sometimes our talks get a bit confused, and so I really want to make sure that everybody knows that it's not being used in the clinical context at the moment, even though that's what we're talking about. Okay, so the concerns. The concerns are not new per se. We've talked about a lot of these concerns when we talked about other reproductive technologies, such as in vitro fertilization and pre-implantation genetic diagnosis, where you'd actually look at an embryo and see if it has, you know, the disease alleles or not the disease alleles, and then you'd only implant those that don't have the disease alleles. That's pre-implantation genetic diagnosis. So we've had a lot of the ethical issues that we've seen with those reproductive technologies are also present with gene editing for reproductive uses or for heritable uses, if you will. So they're not new. So some of the things that I'm going to say may sound generic. There's definitely one that's very new, and I will make sure to point that out. So one of the obvious uses is the use of embryos. I'm saying now in research because you have to imagine that before this would ever get to the clinic, we would have to do a lot of research on it to know about the safety and, of course, the benefits. Would it work? And in that case, you would be creating embryos that would presumably grow beyond the 14-day stage, where we have a law now that you can't let embryos grow beyond that time. We have a whole other webinar about that. But so the use of embryos in research is already contentious and debatable because it challenges the notion of human dignity. And again, that's a whole other webinar, but to make it, to say this quickly, is it depends on the moral status that you give the embryo. If you give the embryo the same moral status as you give any live human, then you might say that you cannot kill it, that you cannot use it and discard it. That's the view of the embryo having the same moral status as a living human. If you say the human embryo actually will have increasing moral status as it develops, then you might say that you can use it in research, and you might say that it's okay if you discard it afterwards. So this, we're not going to answer this. We're not going to answer that question right now. I'm just showing you different ways of thinking about this and why the use of embryos in research is problematic. And if you were going to use gene editing for heritable purposes, for reproductive purposes, there would be embryos used at some stage. And this is a debate that we'd have to address again. And of course, as you know, there are laws that regulate the use of embryos in research where the origin of the embryos matter. You can't create them just for use in research. And what you do with them also matters, and how long you keep them also matters. So these are a lot of issues. The next issue is the one that is really specific to gene editing. And that's basically, what would you do, what does it mean to actively modify a human to actively modify a germline in a human in a lab? There are a lot of laws that prohibit this, and I've listed them here on the slide, and we won't go into that. But the question is, how beneficial would germline gene editing have to be for us to revisit these laws? That's number one. And number two is there a moral issue with having embryos or persons, the cells, human cells, be modified in a lab that will then create new humans? Will that create a problem in society? And we don't have the answer to that yet. There's a lot of debate about that. And this debate obviously should be continued. So the rest of the issues are more generic, and so I will go through them much faster than these first two issues, which I think are much more problematic. The safety looks a lot like the safety issues that I discussed for somatic gene editing. Again, you'd have to wonder how beneficial gene editing would be. And here I give you some examples, which I'm sure you've all thought about, that you could use germline gene editing to prevent severe diseases. It would be used to support reproductive autonomy for persons who want to have a healthy, genetically related child, and it could support the pursuit of scientific freedom. All issues that have been brought up for previous reproductive technologies being used. Of course, when you're talking about safety, you want to wonder about, on the individual level, is it safe for the person that we would be creating using gene editing? But is it also safe for society at large? What does it mean to be systematically taking away a specific genetic variant in the population? And again, this is more an evolutionary genetic debate, which we don't have time to go through, but I raise it here because it's still important to consider this. We don't know what the effects would be. Again, the issue of access to this technology. Will everybody have access? Who will pay? Who will decide on the criteria? Very important for new technologies. The issue of consent is also difficult here because presumably the people who would need this technology would be in one aspect vulnerable if they knew that they had they were at risk of having a child with a disorder, and if they desperately want this cure, then what does it mean for their power of saying no to it if they really want it, which doesn't invalidate the yes that they might give to this consent, but it just says how can we, is there a challenge here of really supporting their yes if they really want it? And of course, is it in the best interest of the child? Presumably, if you're avoiding a disorder, that sounds good, but at the beginning, we still don't know about the safety, and we still don't know how long we'd have to be monitoring these children for. At the beginning, the first children that would have this, presumably, their whole life might be seen as an experiment in some sense. As many, this is not new per se. You could have said this for IVF, for children who were born with IVF as well. As I mentioned for somatic gene therapy, what would be the acceptable use? For which disorders which would you use this? Only disorders where there's no other alternative? Only for severe disorders? And again, this is very much up for debate. In genetics, the stakeholders have not even come to agree on what's a severe disorder for newborn screening. And we have this in other issues in other contexts as well. So this is not easily answered. No, so this issue of where we draw the line, what would it be based on, and who's going to decide are all open questions. As was mentioned in the previous discussion, there's also commercial interest that we have to think about. If people know they can make money off of a technology, then they will try, and that's okay, but that could also distort our view of if we really need this technology or if it's being pushed on us. Because it's new, because it looks good, because it's a high-tech version of potentially something we can do with lower tech. These are just things that we have to consider when we're thinking of using a new technology. And some previous examples where the commercial push was problematic is in stem cell therapies and in direct-to-consumer genetic testing, if you're aware of these phenomena. Finally, my almost last point has to do with the technological imperative, which is, a philosophical concept that says that people might be at the mercy of always wanting to use new technologies, even if they don't really use it. And if you have changed your cell phone to a newer model before your last model stopped working, you might be at this mercy of this imperative. You might be at the mercy of this imperative. And definitely in healthcare, we tend to always want what's new, which could be a good thing, but it's important to question why we want it and why we're using it. Is it a need or is it a want for what's the latest technology? Finally, bravo to EURORDIS, because they're really fulfilling this last ethical and social issue, the need for different publics to be engaged and to get their views on these technologies and the use in humans. Of course, there are different stakeholders, and when you give them a phenomena, they will have a different view on it. And it's important to understand all their different views to see how they will respond to this phenomena if used routinely in the clinic and or in research. So I'm just putting a number of different stakeholders here, and as you can imagine, their concerns and their desires might be different, so it's important to hear from all of them. But of course, there are some challenges which has to do with how do we engage stakeholders at a level where we can really discuss the science. So we need to engage them, we need to educate them, but then we still need to have a dialogue with them. It can't stop with just a survey or just one webinar, although that's a really good start. With this idea, and in gene editing in particular, I have thought of this endeavor. I'm trying to make a project out of it that I'm calling SEED, so it's Stakeholder Engagement, Education, and Dialogue. And we are trying to see how we can engage different publics, including patients, including general public, scientists, experts, etc. But of course, one of the issues here is who's going to do this? Whose responsibility is it to do this? So I'm taking this on as an academic researcher. EURORDIS is taking this on because they want to serve patient populations. But then are they getting the money for it? Are they getting the resources for it? Who is really supporting this? So these are all, again, open questions. And I leave you with this question. What is your role in this? And I also leave you with a bit of more tools or more information if you want to go look at the National Academy's information on gene editing, which is a bit more academic, but potentially still informative. Thank you so much.
Thank you very much, Heidi. That was also very, very interesting. And I just maybe, sorry, I'm not taking that. We already have some questions, so I might not be taking them in order. But just because of your last slide, I wanted to ask the question from Michele and Chris, who mentioned that academics, as you said, just already have a statement that prohibits genome editing for germline use. Do you think that the patient community also need to produce a similar statement?
I think that's very interesting. Do I think that patients need to produce a similar statement? I'm not sure if patients need a statement, but I think patients need to express their opinions, for sure, as much as the experts are, if not more. I think that's definitely needed. The only term I'm comfortable with is statement, because I think you probably can't speak for all patients, or maybe if you can, okay, go ahead. But I think we need as many opinions and thoughts as possible. So yes, definitely any way that the patients can make their voices heard, I think they should.
Thank you. We also had Joseph that made a comment about, I think it was during your presentation, so is this a new ethical issue? And I think you kind of answered that, but Joseph just shout if I'm mistaken. And as the methodology is new, but the use is already known, like gene editing, used by molecular biology techniques already available. So do, you know, those ethical concerns are shared by not just genome editing, but others that, as you mentioned, the stem cells or the IVF or the pre-implantation genetic diagnosis as well?
Right. So they're not, very good question. All the ethical issues are not new. For sure, they're not new. What is very new, though, and what's really troubling us, is the use of this technique in humans for reproductive purposes. So to create a, you know, a genetically modified, if you will, human. That's what's really new. And, you know, we haven't done it yet, and we may never do it, but potentially we have the technology to do it now, whereas before scientists didn't feel that they were that close. So that's really the new aspect here, which I think is monumental.
Thank you, Heidi. We also had Gabor who asked a question. I think it's probably more directed to Fulvio, but feel free to jump in as well, Heidi. How can we use this technique for polygenic diseases?
Well, Fulvio, yeah, go ahead, but I could tell you that it would be very difficult.
Yes, that's what exactly my answer. There's a lot of difficulty. You have to consider that editing precisely one gene is already today quite an effort in terms of building the tools and using them correctly. Heidi mentioned the off-targets effect, which I didn't mention in my presentation, but this is just one example.
Sorry, a little louder if you can, Fulvio. Sorry.
Can you hear me? Can you hear me?
Yes, but it's just a little bit faint. If you could just speak out a little bit.
Okay, I come closer to my microphone. So what I was saying is that our tools may be very precise, but nothing is absolutely precise in nature, certainly not in what humans do. So it's already difficult to find tools that can do precise editing without cutting in places where we don't want to go. So correcting more than one gene, that may be maybe even more difficult. But there are exceptions. As I said at the beginning, with editing tools, we can change regulatory regions. So we can change regions that have an impact on the way cells express not one gene, but 3, 4, 5, 10 genes. So this may apply to some genetic diseases that happens in families of genes that are regulated together. So there are instances in which, not by changing one gene, but by changing the regulatory part of our genome that regulates more than one gene, or more functions of a cell, or ablate completely cells, we can actually address also polygenic diseases. But I would say that today I cannot come up with any practical example of this being a reality. It's a theoretical possibility.
Thank you, Fulvio. We have George who's asking, what are the steps that we need to take before seeing gene editing in clinical use? And can we estimate how long each of these steps will take?
Gene editing is already in clinical use. The example I made of AIDS gene therapy, it's already a clinical trial. It's ongoing at the University of Southern California. There are patients treated. So it is in the clinics. It's not as widely used as what I called classical gene therapy with viral vectors, simply because it's newer and we don't know exactly how to use it safely. But there are things that are moving into the clinics, and they are moving fast. I do expect that changing stem cells, so doing ex vivo gene therapy, as the example I made for AIDS gene therapy, is going to be the applications that is going to be used in the short term. In vivo editing, so editing something in the entire body, it's much more difficult. We don't really have the tools to do that today, but I'm sure this will come relatively soon. I do expect that between 3 and 10 years, this will become routine forms of gene therapy.
But just to say also that, so for somatic gene editing, they're following, you know, the same, not the same exactly, but you can think of clinical trials. What do you need to get a new drug onto the market? So they have this modified version of clinical trials, if you will, that they're following for clinical gene editing, for chromatic gene editing. For germline gene editing, potentially that's what the question was about. And, you know, again, we're going to depend on the, we would depend on something that looks like the clinical trial protocols. But of course, there's going to be a lot more discussion before that happens, for sure. If the last 24 months are an example, the steps, you know, it's not going to happen smoothly. There's going to be a lot more discussion, which I think is a good thing.
Thank you both. Maybe we can have one last question from Deb. What is an example of an off-target event in human disease treatment? Do you imagine a possibility?
So I use the term off-target. Oh, go ahead, go ahead.
No, please.
Well, I just want to say that I use the word off-target event to mean, you know, a side effect, a negative side effect. So you could, two things. You could, you know, target the gene that you wanted to target and everything went well, but there's still a side effect. I don't know, an epigenetic side effect, or any effect due to the machinery to the tool that was used. Or you could also, you know, have an off-target event, so you actually edited the wrong, or, you know, edited where you wanted to edit, but also at other places, and that caused downstream side effects. So I just meant any physiological side effect. Fulvio, maybe you could give us more detailed.
No, that's perfectly right. Off-target means, and the way we use the word is to indicate the unwanted effect of our precise forms of editing. So what we think is precise. So as I probably mentioned, what we use today is, in the case of CRISPR, for instance, is an RNA molecule which is complementary to a sequence of DNA. And this is what we do to direct the cut to a specific region of the genome. But the genome is enormous. So there are regions that are similar to the region that we want to target. And sometimes our precise molecular tool can do something very imprecise, so cut somewhere else. So this will be what we call an off-target effect, so outside the target. And sometimes we cannot predict it, sometimes we can predict it and check for it, but this is in the, as Heidi said, in this what we put in the side effects. Just to reassure who asked the question, the forms of gene therapy that we use today and have an enormous number of off-targets events. So we use vectors that go anywhere in the genome, and in most cases randomly. So what we use today in patients, and we know it's relatively safe, goes everywhere. So we know that by doing something that we think is efficacious, we potentially are touching pieces of the genome with potentially terrible unwanted consequences. And actually, we have seen this in gene therapy. So for instance, in some clinical trials, leukemia emerged as an unwanted side effect of an efficacious treatment. So the vector that was used to correct the genetic disease in the inherited immunodeficiency landed into an oncogene and caused a transformation. So gene editing is supposed to be, and we hope it will be, much more precise than that. So off-targets worries us, of course, as scientists today. We want to limit off-targets as much as possible, but we are moving from a field in which there is no target. So we basically put things randomly into the genome into a field in which we go precisely, but sometimes not so precisely. So I see this as a tremendous progress in terms of risk-benefit with respect to the gene therapy that every day we are using.
Thank you very much. Just one last question, and then we'll probably stop after that one. From Danielle, is there any economical issue hidden behind, in the sense that gene therapy can correct default, but which can be costly to deal with the entire life? What are the health authority on that aspect? Are they interested to save money by treating patients? What do you think?
I'm not sure if I understand this question correctly, but I will rephrase it and rewrite. So, of course, you know, in reducing the burden of disease, we want to reduce suffering, human suffering, and of course there is a cost-benefit calculation based on actual monetary cost that is done usually by healthcare systems to see if they want to include, you know, or reimburse a technology or a treatment or a medication. So, of course, there could be a cost. There usually is a cost evaluation made for all the therapies and medications used in a healthcare system. So is there a hidden economic issue here? I would say no, not with respect to wanting to save money and save lives. I think that's very transparently open. So maybe, Virginie, let me know if I've understood the question correctly. I'm going to reread it here. What are the health?
Hello?
Yeah. So.
I don't know, Danielle, did Heidi answer your question?
Can you hear me?
Yes, we can. Go ahead.
Well,
thank you.
No, I was, because we discussed ethical issues and the efficiency issues of this thing, but the public health authorities are always looking for saving money. And I think that there might be some policy issues behind this technique, because they can say, okay, we will save a lot of money by pushing this therapy to reduce the burden of for the public health system of the diseases. So I just want to highlight this and putting this into a question.
Well, that's the case for a lot of new treatments and new technologies, but for now, for the somatic cell therapies that Fulvio has talked about, in many cases, there are not a lot of other alternatives to treatment. So, you know, for HIV, there's, of course, very costly drugs that have been efficient for some people, but not for all. But I'm not sure the push for somatic cell treatment is necessarily involving any hidden agendas there. But indeed, you are correct that if they find a treatment that's cheaper, they often try to go there. We like to think that they also do that, if it's an efficient treatment that is best for the patient, of course. But I think you're right that we need to keep considering how much the economic factor plays into any decision.
If I can add something to this discussion, which is extremely interesting, and actually it's a discussion ongoing between the industry, the academia, and the regulatory authorities and the governments. The model that governments use today is it doesn't matter how expensive a new treatment is. And in this, gene editing or gene therapy are exactly equal to any new forms of treatment. So there are very expensive drugs to treat cancer, for instance, and they are on the market. So what the health authorities normally consider is, okay, we pay for this very expensive drug, but what do we save by not having the burden of keeping these patients alive or things like that? So think about, I don't know, muscular dystrophy, an enormous cost for individuals, families, and the society for caring for these patients for 10, 20, 30, 40 years. So if there is a treatment, unfortunately today we don't have it, that it doesn't matter how expensive, but millions, but can save 40 years of continuous therapies and 40 years of suffering for that, they will consider it. On the other hand, a very expensive treatment that doesn't really change the natural course of the disease, or that doesn't really have an impact on the disease, will not be reimbursed. So I think that the health authorities, of course, they want to save money, but they are well equipped, at least in most European countries, to consider the risks and benefits of paying high prices for new medicines and putting them in a context, in a global context of the economy of the health system. I'm optimistic.
Thank you very much. Thank you to both of you. I'm sorry, I think we could keep on going, but unfortunately we're going to have to finish the webinar. So I just wanted to say big thank you to both Fulvio and Heidi for giving their time and providing us with really interesting presentations and information. And also thank you to everyone who joined us. I just wanted to confirm that I will be sharing the webinar afterwards by a recording of the webinar. So for those as well who did not attend, and you'll be able to share with your patient organization. And it will also be added onto the webinar section of on the EURORDIS website. So thank you again, everyone. Thank you, Fulvio and Heidi. And,
thank you.
Thank you.
Thank you very much.
Everyone.
Merci, au revoir.
Bye.
Ciao.